Chester Whitley, who's going to have a talk on pluripotent stem cell gene editing for Pompe disease. And Chester Whitley is a professor of human genetics at the University of Minnesota, and he has a long history in lysosomal storage disorders, in bone marrow transplantation, in MPS. And you're also the organizer of the World Conference, and that's a very important conference that's yearly held in Florida, isn't it? Yeah.
Well, thank you very much. I guess I can sit down now. That's
please
great.
Commence.
Really, I want to thank everybody for coming to here. This is research for me, and research is always what you've done in the past and what you want to do in the future. So I will walk down that path. I don't want to overpromise things in the abstract that would imply that maybe we've done some of the things we haven't quite yet done. But we did apply for financial support to do some of the things we want to do from AMDA, and Tiffany House and members said, well, why don't you come and talk at the meeting? So we've not been funded for anything yet, but I'd like to show you what we've done and what we'd really like to do. So in context, where are we with Pompe disease? In Minnesota, we've been screening for newborns for about 3-4 years now. I'll mention briefly the NIH-supported Lysosomal Disease Network, and I'll also talk about the impact that's had in clinical directions for our research and in the future. How do we move toward really adequate therapy and the role of the FDA and economics if we've done that. So in Minnesota, we have 3 tertiary care centers that see all the newborns who are products of newborn screening. We get about 68,000 births per year in the state of Minnesota. At the top, you see the State Department of Health that does that screening. They're very collaborative, they're very aggressive, they're very helpful. And they'll send out a notice to immediately the clinicians that may be involved. And so us clinicians at the Mayo Clinic, the University of Minnesota, and Children's of Minnesota have to be on board ready to treat those patients immediately. And that is our Minnesota Pompe Disease Consortium, led by Nishitha Pillai. And these are some of the people involved. We're trying to do this in an organized fashion. Sometimes such endeavors as newborn screening are top-down. This is a little bit more grassroots, but everybody was so collaborative, and we can do some things as a consequence. One of the important things we discovered we've published, and that is that when you diagnose a baby with Pompe disease, the families are relieved when they find out it's not affecting the baby right now and there's no treatment. And that might be a false positive. With more concern, it could be late onset Pompe disease. And I personally think that's one of our biggest shortcomings, losing those patients to follow-up. We don't want to see people who are 10 or 15 or 50 years of age not recognizing they had late onset Pompe disease when they were a baby. And so we're doing everything we can to think about how can medical records maintain that diagnosis so that 3 or 4 generations of physicians taking care of that child have forgotten the diagnosis. It's amazing. It was a special problem because a number of our patients who were lost to follow-up were immigrants, Somalian, Cambodian folks, who really did not want to be involved in our care. They thought their child was doing fine, and that's the message they're going to carry forward to their primary care doctors if we never see them again, which is a grave concern we have. During that process, one of our first patients was taken care of by Dr. Jeanine Jarnes, and maybe you've heard this story before. But this was a newborn baby who was positive. At age day 8, she was diagnosed, seen in the hospital. She had biventricular cardiac hypertrophy and the abnormal biomarkers CK, her Hex4, troponin, and transaminases, and mildly reduced muscle tone, no macroglossia or hepatosplenomegaly at that point, or feeding difficulties. And her genotype was entirely consistent with the clinical picture of neonatal lethal Pompe disease. So we could approach this several different ways. Of course, we did CRIM testing. The baby was CRIM positive, and ERT was started right away. Of course, we have to go through the insurance policy, approval and that sort of thing. But what was remarkable is Dr. Jarnes, bringing experience from hemophilia and cancer, took a very aggressive approach and treated the baby with 400% of the label-approved dose divided into 3 weekly doses. So this was not continuous enzyme replacement therapy, but it's as much as you could do under the circumstances. So this is that little baby now at 12 months of age, and people will wonder, how is that possible to give 3 doses per week? Well, here it is, folks. She's carrying her own IV bag on her back in her backpack, and you see here at this age, really getting up. And when a 12-month-old is at this point, they should be walking. Well, she's not only walking, she's carrying her luggage with her. She's carrying her IV pump and her IV bag there. So this little lady has progressed on. Dr. Jarnes has followed her very carefully and continuously, and I hope I hit the right back button here. There we go. And I want this to go, and it's not going to go. What do I do here? Oh, it's coming on. It's coming on. So you'll see her in the distance here, and she's going to get closer to us in a minute. But she's really maintained normal motor function as well as intellectual function. Here she's just, enjoying what a normal kid does. And you wouldn't expect too much, limb girdle strength to do swings. But she'll get off the swing here and show us a little bit more in a few minutes. But this child, by very rigorous testing, has no physical or cognitive disorder that we can identify, and her biomarkers are normal. And we're very optimistic about her. And there she's got her pack still. I don't know if she's getting infusion right now, but she's become very accustomed to going off to school and getting not continuous, but very, very frequent enzyme replacement therapy. And I think that's what's keeping her in great shape. So thanks very much. But let's go way back in history here. Hurler syndrome was identified in the 1980s as a target for bone marrow transplant experimentally. And with Jack Hobbs in London doing the first bone marrow transplant for Hurler syndrome, we had evidence that we could transplant enzyme into a patient with a disease, in this case a hematopoietic stem cell transplant, and treat the disease. And so one of my roles early in my career was to spend 10 years doing ongoing long-term systematic evaluation of those patients. And frankly, to my surprise, we did learn that we could protect the brain against deterioration if we do a successful transplant. And that really set the stage for a lot of the other things we were doing. Of course, in the global, Roscoe Brady in the 1990s was actually doing the first INDs for enzyme replacement therapy, and the rest is history, including treatments for Pompe disease. Well, now looking forward, let's talk about a few more things. And I'm going to talk about gene editing in general. The NIH has caught a lot of attention by putting a lot of money into gene editing. Our experience started with Sangamo, a corporate-sponsored in vivo zinc finger nucleotide gene editing. And here you can see how that works at the cellular level. This is a piece of DNA. Let's say it's intron 1 of the albumin gene in the liver. And in the lower left-hand side, on the top row there, you see one zinc finger nuclease binding to DNA, and you see that dark blue scissors there cutting the DNA. A second zinc finger nuclease is cutting on the opposite side, and together those two zinc finger nucleases bind to one specific site. In this case, in intron 1 of the albumin gene. The DNA is severed there, and then a third molecule comes in, all delivered by AAV viruses, by the way, to insert a therapeutic gene of choice. Could be for Pompe disease, could be for Hurler syndrome, in that place. And so from now on, the albumin promoter in the liver thinks it's making albumin, but it's actually pumping out whatever that therapeutic gene is supposed to be making. So we were the ones that proved to Sangamo that their mechanism actually worked in the MPS 1 mouse, in the MPS 2 mouse, Hurler syndrome and Hunter syndrome. And here's how it works. You can see that the zinc finger nuclease is encapsulated there. Notice the red letters 3, not 1, but 3 different AAV vectors. They're required to be infused simultaneously. They go to the liver. A small number of liver hepatocytes think they're making albumin. They're actually making an enzyme, a therapeutic enzyme, that goes into the blood, that is distributed to spleen, heart, lungs, kidney, just as though that was a bone marrow transplant, with all the problems that go with it, the limitations, but some therapy. But emphasizing here the 3 AAV vectors, that's a big load of drug to make. AAV vectors are not cheap. And if you look at the stoichiometry of it, it's a problem. This is actually Brian Mado, a 44-year-old gentleman out in Oakland, California, who was the first human being ever to have in vivo gene editing. You can see there with his wife, they just finished his first infusion of 3 AAV vectors simultaneously. Now that clinical trial continued on, and everybody got through it, no problems, but there was insufficient clinical response. So what's going on with that? Well, I think stoichiometry is the issue. And when you think about it, if the probability, this is purely hypothetical, of injecting one AAV vector into the bloodstream and having it wind up in a hepatocyte where it needs to go, let's just say that's a 10% probability. 1 out of 10 hepatocytes will get one of those AAV vectors. But if you're delivering 3 of them, and all 3 have to wind up in the same cell, that's adverse stoichiometry. You've got only 1 out of 1,000 cells getting all 3 AAV vectors at the same time. And I think getting that dose of enzyme is insufficient. They proved it, unfortunately, and that approach has been abandoned for the time being. However, during those studies, people in my lab were becoming very aware of CRISPR and its possibility to do a very, very similar kind of thing. So they, if you will, invented what we call the PS gene editing system. Here you see a similar diagram of the albumin gene. Cas9 is coming in delivered by an AAV vector, and it's inserting in a site, a unique site, in the albumin intron 1. And it's inserting in a therapeutic gene. And we'll try a few genes here, and I'll show you how that works. There's different kinds of recombinant technology going on here, or recombinant events. But to the point, this is what the new kind of gene editing in the same area is. So in the yellow there, you see the DNA and the guide RNA in one AAV vector, Cas9 delivered in the second AAV vector, so 2 AAV vectors are given to the mouse intravenously. The therapeutic gene goes into hepatocytes. Those hepatocytes secrete from the cells, not albumin, but a therapeutic enzyme. And we've therefore infused the stoichiometry of that. So now instead of getting just 1 out of 1,000 cells genetically, dare I say, call it corrected, at least becoming a factory, you're up to 1 out of 100 cells. You've got a 1 log-fold improvement in the stoichiometry just by packaging things differently. So in mice, we've demonstrated this in the Hurler mouse. We've demonstrated it in the Sandhoff mouse, primarily brain diseases. And by the way we treat the Sandhoff mouse, we're simultaneously creating an agent that could be used for Tay-Sachs disease. And then we've also done this for a GM1 gangliosidosis mouse, which would not only treat that disease, that brain disease, but also Merkel syndrome type B. They're both missing beta-galactosidase. So previous studies showed that high and constant levels of enzyme given systemically do cross the blood-brain barrier. That violates one of the central dogmas of neurology. You can't give a drug, a large protein intravenously and get it in the brain. But I left out one slide here that talks about 14 examples of that in animals. You can look at an article by Lund et al. That came out about 6 months ago where in human bone marrow transplant patients that's been demonstrated when they're getting IV drug. But going on, we want to look at what it would take to enable a human trial, the IND-enabling studies of PS gene editing. And I'll give you a taste for some of those things that we've done. A lot of them are based on the MPS1 mouse model, but I'll show you some other models. Here, for example, we look at Kaplan-Meier survival, fear conditioning, a learning test, a POL test that also looks at strength, as well as cognition and rotarod tests. In all those cases, the treated animals do better than the controls. If we look at the pathology, we look at the liver and the brain. In the liver, on the left, you see a normal liver in one of these mice, the untreated Hurler mouse in the middle, and then to the right, the mouse liver who's had a PS gene editing treatment. If you look at the brain, one is actually seeing enzyme effect in the brain. We're not giving AAV vectors to go to the brain. We're not expecting the enzyme to go to the brain, but if you give enough enzyme intravenously, it goes to the brain. So in terms of its safety, we're not seeing vector-associated problems in the mice. We're not seeing other toxicity, no evidence of tumors. So, those are the kind of things you'd want to know about before you go to the FDA. We're looking for antibodies against the enzyme, antibodies against the Cas9 enzyme, antibodies against the AAV vector capsid itself. So those all look pretty good in mice. Always the caveat, these are in mice. We look for cytokine biomarkers, and there seems to be a lack of cellular response too. So these are safety markers one would want to use, as well as looking at liver function, enzymes, AST, ALT. And importantly, remember, we're co-opting or taking away some of the mouse's normal albumin production, using it to make enzyme. So what happens to the albumin levels? Well, they stay pretty good. They're normal. There may be a transient dip. So in summary, we can show this method is highly efficacious in mice. I want to emphasize that. And we would like to do more with this. Importantly, most people don't believe that a high level of enzyme goes to the brain if it's coming from the blood. And in fact, there might be some reasons to augment the amount of enzyme going to the brain or other places like the muscle. So one thing that we've done is looked at the hexosaminidase enzyme for Tay-Sachs-Sandhoff disease. Here in white is a variant of that enzyme. It turns out, for those of you who understand this disease, you have to have an alpha subunit and a beta subunit to make hexosaminidase A. And you can't fit that in an AAV vector. It's too big. The payload is too big for an AAV vector, so people have not even pursued that by and large. But we've taken what we call hex-O, and there's some other variants, other people that have done similar things, and made one protein that can serve as both the alpha and the beta unit. So it makes a homodimer, not of the alpha unit or the beta unit, but the hex-O unit. And you can see here in this molecular modeling, the active site for the enzyme to break down the GM2 ganglioside is in the middle. Over to the green is where the enzyme binds the GM2 activator, another component needed to break down gangliosides. Over on the right is a fusion part. We're adding something that can maybe make this enzyme get picked up and go across the blood-brain barrier better. In fact, here's a different model using a different fusion in red. And a third one, this is the anti-insulin receptor antibody that's being used now for some commercial drugs that are aimed to be given intravenously but go across the blood-brain barrier. So we didn't have to do all these experiments in a test tube or a mouse. This is just computer modeling. But it helped us to pick out one that we like here. We've actually picked out several based on their free energy. And Hexo there at the top has the greatest free energy. It's going to be the most stable molecule. So that one was used in all the subsequent studies. And here in blue, that blue dotted line, the Kaplan-Meier, shows that that does better than all the other fusion proteins we made. Probably it's doing better in the brain. And we've done some testing with that. So where would we like to go? Look at what we've done to the stoichiometry already. If you start with the same number of vector particles, be it AAV vector or something else, and you have to give 3 of them, you're only getting 1 out of 1,000 cells if the underlying rate of getting something that's injected into a hepatocyte. If you go down to this system, 2 vectors, hey, you've improved by a log fold. And that's time up for me, so I'm going to end by saying if we go to 1 vector, we've improved the efficiency one more. To the point, Peter Marks is director of the FDA Center for Biologics Review and Evaluation. And last February at the meeting, he was looking at the problem of how do we get all these drugs through FDA, which is another way of saying how can we get them there faster and cheaper. And he pointed out this article, this legislation that says if a single entity makes a plug-and-play platform, the FDA won't have to review everything every time. It will only review a small portion of those things because they know the way you made it, the reagents you used to make it, they're pretty much all the same. So this is legislation which he's exploiting, and he will give us faster approval of drug if we do that. And so the first thing he mentioned was, on the left here, CRISPR-Cas9, and especially lipid nanoparticles. And so that's what we'd like to do next. Thanks for your attention, everybody.